So I have the very great pleasure of introducing Dr. Doug Wallace, who has been really a founding father of mitochondrial DNA causing human disease, so has a lot to share with us. Is at the Children's Hospital of Philadelphia, and is today discussing with us haplogroup compatibility and how mitochondrial DNA can influence traits beyond disease.
Oh, great.
I think your mic's off. Maybe push the button. Try. There you go.
I was asked specifically to talk about haplogroup compatibility and how mitochondrial DNA can influence traits beyond disease, and I'm specifically going to talk about that topic. One of the things that has been suggested is maybe the mitochondrial DNA is not all that important. I'd like to make a very strong statement that I don't agree with that. The mitochondrial DNA does only code for 13 proteins, but there are 7 of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5, and these proteins are critical electron and proton carriers for this system, which is the very heart of making energy for your cells. So changes in any one of these proteins is very, very important. Now, the mitochondrial DNA is a circular molecule, 16,569 base pairs. ND 1, 2, 3, 4, L4, 5, and 6 code for the proteins of complex 1, cytochrome B, the protein of complex 3, CO1, 2, and 3 for complex 4, and ATPase 6 and 8 for complex 5. And as you well know, it's maternally inherited, and we have this process of heteroplasmy and this random segregation, as Dr. Shoebridge mentioned. So there are really 3 classes of mitochondrial DNA mutations that are relevant to disease. One is the one we've been talking about, which is recent deleterious mutations that occur in the maternal line, and you heard about the 14459 from Dr. Morais. But there are a variety of different ones. Mutation in the tRNA leucine gene is the MELAS mutation that you've heard mentioned. We're going to mention a little bit about the tRNA lysine gene, which predisposes to late onset Alzheimer's and Parkinson's disease. Glutamine, I'm sorry, tRNA lysine gene as myoclonic epilepsy, many tRNA mutations, and many polypeptide mutations that are involved both in inherited disease and also predisposition to cancer. So these are the maternally inherited diseases, but there are also ancient polymorphisms. So this variant, which we call macrohaplogroup L in ND1, is found in 3/4 of sub-Saharan Africans. Variant H is found in about half of Europeans. Variant A, B, C, and D arose in Asia, crossed the Bering Land Bridge, colonized the Americas. And these mitochondrial DNAs were the ones that were, these variations were used to trace human origins. We showed that the mitochondrial DNA arose in Africa about 150,000 years ago, radiated in Africa until 65,000 years ago, where 2 lineages, M and N, arose. And N and M are the only 2 mitochondrial DNAs that left Africa and colonized the rest of the world. N went into the temperate zone and gave rise to the European lineages, H, J, T, U, U, K, and V, I, W, X. M stayed in the tropics all the way down to Australia, and much later moved into the Central Asia to give rise to lineages C, D, G, and a whole bunch of M's. And N also moved into Asia. And then much later, C, D, and A became enriched in Chukotka, crossed the Bering Land Bridge 20,000 years ago, and migrated into the New World. And we feel that these changes in mitochondrial DNA as women migrated out of Africa into the rest of the world were in fact adaptive mutations that changed the mitochondrial function to allow people to live in the different climatic and environmental contexts that they were found in. This is just one of many examples. This is a variant that rose about 15,000 years ago in the European population. It's an A to G variant in this tRNA glutamine gene. It's found currently in about 0.4% of all European individuals. But if these people live to about age 70 to 80, they then account for about 3% of AD, Alzheimer's disease, 5% of Parkinson's, and about 7% of Alzheimer's Parkinson's Parkinson's disease. So this leads to an interesting question. How could a mutation that is bad become polymorphic in the human population? And the reason is that our ancestors didn't live past 50 years. So these kinds of effects, these very late onset effects, were irrelevant. But we think this variant actually increases mitochondrial reactive oxygen species production and actually is advantageous against infectious disease. So in fact, there's a lot of what we call antagonistic pleiotrophy. A variant that's good in one part of life is deleterious in another. So it's not at all easy to decide what is a good and what is not a good mitochondrial DNA. And these are just some associations that have been made with the European mitochondrial DNAs for neurodegenerative diseases like Alzheimer's, Parkinson's, macular degeneration, migraine, headache, psychiatric disorders, stroke, diabetes, cardiovascular disease, metabolic syndrome, sepsis, IgE levels, asthma, AIDS production, anti-AIDS, osteoarthritis, aging, cancer, but those all sound negative, but these same mitochondrial DNA lineages radically affect athletic performance. So what is bad in one context could be very good in another context. Okay, so one of the things that Dr. Cohen, who's here, did as a pioneering experiment is he wanted to reactivate eggs of older women. And one idea was that the cytoplasm of the egg had become damaged, and if you added new cytoplasm, maybe the fertility would be increased. So his pioneering experiment was to take up the sperm by ICSI, take about 20% of the donor oocyte, inject it into the older woman's egg to give you then a mixed egg, which then created a child, and some of these children were heteroplasmic because, of course, there were two different mitochondrial DNAs. So we were interested in seeing whether that might have any health effect, and we took advantage of work that originally started, as Eric said, with Hauswirth, but then Eric Shoebridge pioneered in this work of mixing two normal mitochondrial DNAs in the same cell. We used a little bit different approach. We took a female embryonic stem cell from a 129 mouse, cured it of its resident mitochondrial DNA with a drug called rhodamin 6G. We made a cell that had a different mitochondrial DNA from an NZB mouse. These are both normal mice. Removed its nucleus, took the cytoplasmic fragment, and now mixed the two mitochondrial DNAs into the primordial germ cell, put it into a blastocyst, put it in a foster mother, created a chimeric female, and bred for the transmission of the dominant Goodylocus, picking up the heteroplasmy. So this just shows that in the in the founder female, we have both the NZB and the 129, rather similar to what Carlos mentioned. And you can see that the female transmits this heteroplasmy to all of her daughters, and her daughters transmit it, whereas the males do not transmit the heteroplasmy. Not surprising. So we then back-crossed this heteroplasmic cell line onto the same nuclear background till we had an absolutely uniform nuclear background. There's about, 91 specific variants between them, which, by the way, would be the difference between, say, a person in South Africa versus a person in Europe. Okay, so we then had a heteroplasmic mouse, and then what we did is we bred that mouse to segregate back to either pure 129 or pure NZB, or to have heteroplasmy. And that will then allow us, on the exact same nuclear background, to compare whether there was any phenotypic effect of the heteroplasmy. And when we did this, this is the 129 mouse genetics. And these mice are active at night, and you can see at night they're very active, but in daytime they're rather less active. And this is the NZB, exactly the same. But when we looked at the activity of the heteroplasmic animal, you can see that it was much less active, about 30%. It also had a slower, slightly less respiration rate, and that was compensated by a lower diet. When we put the animal into a stressful environment, the 129 and the NZB animals quickly learned that the environment was not threatening, and they calmed down, but the heteroplasmic animal was out of control. So it's hyperexcitable, but also depressed. We then asked, will the animal have a different learning capacity? So we use this open field system called a Barnes maze. We have many holes around the outside of the platform. We have different colored patches so the mouse can orient by sight, and we have a little black box there because the mouse likes to hide. So then you put the animal in the middle of the field and you watch what it does to try and find a place to hide. So eventually it will find the right hole and jump in, and you can time that. So this is what we're calling latency. So if we look at the 129 and the 129 and the NZB, at each successive day, they learn to find the hole faster because they're learning the colors. The heteroplasmic animal is a bit slower. But what we did then is take the animal out of that environment, just let them rest for 2 days, and then tested them again. And the 129 animals immediately recognized where they are and jumped in the hole. The heteroplasmic animal also recognized where they were and jumped in the hole, but that means the NZB, but the heteroplasmic animal had completely forgotten everything it had learned. So just having heteroplasmy with exactly the same nucleus caused an effect on their behavior, their ability to deal with stress, and in fact their learning capacity. So I think we do have to be a little bit concerned about heteroplasmy. Now this is a study not from our lab, but one of the ones that one of our speakers mentioned earlier, from Roberto, I guess. But what they were really doing is they took a mouse with, they called H lineage and a mouse with N lineage, and they crossed it so that ultimately they put on the H lineage the mitochondrial DNA from N, and on the N lineage the mitochondrial DNA, I got that wrong, that should be N, mitochondrial DNA for H. And then they just looked at a whole series of behavioral functions. And you can immediately see that H is very different from N on this particular, this is a radial maze like we had. But if you now exchange the mitochondrial DNA, you actually get a rather different result. This is another kind of test, and you can see that the H did very well. The H with N mitochondrial DNA did better. The N did terribly, and the N actually did better when it got H. And here is the Morrison water maze. You can see that H did extremely well in learning how to find this little platform so it could get out of the water, but all of the different lineages were much less effective. So just changing the mitochondrial DNA of 2 perfectly normal mice, just switching them on the background, changed significantly the phenotype in the neuropsychiatric parameter of the wild type. So just to close then, these are some thoughts that I have. Now, first of all, I am in favor of the mitochondrial therapeutic for women with severe mitochondrial DNA disease. And I believe the reason for that is I've seen pedigrees where the woman harbors a mitochondrial tRNA mutation and has had 6 or 7 reproductive failures in a row. And these are very severe ones where they've lost a pregnancy, they've had a stillborn, they've had a child born that died of a terrible death, and they want to have a child, they keep trying, but it's always very bad. So this is a huge genetic load on the family and on the children. So for those kinds of women, I think we need to try and find ways to help them with their reproductive future. The reason, though, as I said, that this is justified is because the negative outcome is so severe. I think, though, it would be very important to keep in mind the heteroplasmy level, which Jeff had talked about in his first slide. I would like to suggest that it isn't completely neutral to switch the mitochondrial DNA and nuclear DNA. Not that one is better or one is less good, it's just that we don't know. And so I'm not saying that the Japanese-American young woman isn't a perfectly fine human being, but she might have been a different human being in a different context. So one of the things we might imagine is to actually keep in mind the haplogroup and use that to our advantage. So if the woman was heteroplasmic, then it's a new mitochondrial DNA mutation, and she probably has on the maternal lineage a relative that was still, existed or was the transmission before the mutation occurred. So hypothetically, if she donated her oocyte, this heteroplasmy could be eliminated with exactly the same wild-type mitochondrial DNA. Or, failing that, we certainly have the capability of genetically matching haplogroups, just like we do HLA matching for organ transplantation. So why can't we take advantage of this opportunity? One of the things that has been mentioned, but I think is very important to talk about, if we go this route, which I, again, favor, Then the question is, what will this technology be used for? And in the United States, as it has been said, we have a very poor regulatory system for this kind of reproductive system. And a lot of people that I've talked to say what's really going to drive this is the financial advantage for advanced maternal age. So women with advanced maternal age are going to believe that their cytoplasm is somehow defective, and they're going to use nuclear transplantation to revitalize their cytoplasm and have a healthy child. Now, I am not formally opposed to this. However, until we understand what this risk-cost-benefit analysis is, what the risks might be, I think we have to avoid this, because this will be a few dozen women, but this will be thousands. And if there is a long-term effect, say 20 or 30 years down the road, we won't know that if we transition too rapidly, and then we could get into a significant problem. Thank you.